Okay, so before we get started with the new material today, I want to take a second just to talk about taking notes in this class and how to kind of tips on how to succeed. So the first part of the class was largely a lot of calculation. So we did a lot with probability and pedigrees and the percent chance that somebody would be afflicted with a particular disease. For the remainder of the course, it's going to be much less based on calculations, and there will be a little more material just to remember. So, I thought I would just take a second and just tell you a little bit about how I would go about taking notes for this class. So whenever I was an undergrad, I think I've mentioned before, I was a chemistry major, and one of the things about being a chemistry major is that it can be tough in a lot of ways, but there's not a lot of material to memorize. So I didn't really get very good at processing large amounts of information until I went to graduate school. So it took me a very long time to figure out how to really take notes very effectively. And you've got an additional challenge in that you're not really taking your own notes to a large extent because I provide you with notes. So what I would suggest is, as you go through your notes, is to create your own set of notes rather than just relying on my PowerPoint slides. So what do I mean by that is that I would suggest that you go through the notes after the class is over and try to condense the notes from the course down to maybe about 1.5 to 2 pages of notes per lecture. And this is going to do a lot of things for you. It's going to allow you to condense those notes, going from 30 PowerPoint slides down into maybe 2 pages of text that you can use to study for the exam. But also, you'll find that it's going to help a lot in letting you assess how well you understand the information. Because if you can condense a slide down into one concise sentence, then it shows that you have a pretty good understanding of it. But if you get to a slide where you try to summarize what it is in just a sentence or two, and you find that you can't, then that's probably a pretty good indication that this is where you've got a bit of an issue and you need to go study some more. So just to kind of give you a real brief example, I'm going to show you a few slides from previous lectures, the ones on genomics where we talked about identifying genes in the genome, and just give you an example of how I would condense these things down into a set of my own notes. So we talked about identifying genes, and we talked about 3 ways to do this. We talked about ESTs, where we sequence cDNA libraries and just kind of get this little bit of a tag on them. Talked about the fact that this won't identify non-coding RNAs because of how reverse transcription works from these oligo-DT primers. We talked about EST sequences, using computers to find these sequence tags that we've created in the genomic DNA. And this will be able to align these ESTs with the genomic DNA, identifying where this messenger RNA is, and therefore there is a gene. And we talked about the limitations, that we won't find non-coding RNAs, cDNA libraries only find the genes that are expressed in those particular tissues, and there's always problems getting rare messages. And I gave you an example of this as to how these rare messages are very hard to find in a sea of more abundant ones. Now for me, I would distill this down into about this. So those, 4 or 5 slides, 3 ways to identify genes. We talked about ESTs, sequence random cDNA clones. cDNA clones are messenger RNA that's converted to cDNA. On the upside, everything you sequence is a gene. Downside, you'll miss non-coding RNAs and you may miss rare messenger RNAs. So for the next part of this lecture, we talked about gene prediction programs and what they look for. Look for things like open reading frames. Things like stop codon should come up fairly frequently if it's not a real open reading frame. And if you get DNA sequences that go on for many, many bases without a stop codon, then it's a good indication that this is a gene. We talked a bit about how codon usage varies, so in addition to just looking for open reading frames using these different codons, different organisms will have preferences for different ones. So again, took a couple of slides down into just a couple of lines. Gene prediction programs look for open reading frames. Stops normally occur about every 20 codons, and just a reminder that codon usage is an issue. Talked about comparison to other genomes. If you have 2 genomes, you can align them to each other in those things which have similarities. are things which have been conserved over millions of years and therefore are probably important and are likely to be genes. And just kind of comparing genomes, important sequences are likely to be conserved, junk, things that are not genes will not be conserved. So this is probably like close to a third of a lecture anyway, that I've gotten down into probably not much more than about half a page of notes. So again, I think this sort of thing will help a lot, both in terms of giving you a nice, concise packet of information that you can study when the exam comes, but also just the act of generating this sort of thing is going to make you think about things and make you think about how to say things in your own words. And it, like I said at the beginning, if you have difficulty saying things in your own words, then this is probably a sign that you need to go back and make sure that you really understand that material. So what I want to move on to today is actually cloning genes. So this is one of the things that we care most about. Many, if not most of you in the class have aspirations of medical school and things like that. So actually identifying the genes that are associated with disease is a big goal of modern genetics. And I want to talk a bit about the basic approaches in this lecture and the next lecture to identifying those genes that cause diseases. So, as we talked about last time, we're kind of changed our definition of what alleles are. So, historically, alleles were things that caused alternative phenotypes. So, back in when we talked about Mendelian genetics, alleles cause either purple flowers or white flowers. They cause round peas versus wrinkled peas. But now that we have DNA sequences, that we have these sequenced genomes, alleles don't have to be things that actually cause differences in phenotype. They can be anything that is a difference in the DNA sequence. So most polymorphisms, or differences in DNA sequences, actually don't occur in genes. So not surprisingly, the sequences that are outside of the genes are the ones that are often the most flexible in terms of being able to be changed without any significant penalty to the organism. So any sequence differences can be called different alleles, regardless of whether they're in a gene or whether they cause a phenotype. So we talked a lot about genes. One of the other terms we're going to talk about a bit is a locus. And a locus is a more general term that just refers to any specific location on a chromosome. It can be in a gene or it can be in between genes, or it could be in an intron or an exon. So basically what we're looking at now is if we have two DNA sequences, these could be the two sequences in one individual. One came from the maternal copy of the chromosome, the other came from the paternal copy of the chromosome. As long as there's a sequence difference, then these become two different alleles. These might be in a gene, they might not be, they might cause a phenotype, they might not. But as long as we can see this difference, we can detect that and we can tell this chromosome from this chromosome, therefore these are different alleles. So, types of polymorphisms, we talked a bit about these before. The most common are single nucleotide polymorphisms, or SNPs. There's about 100 million in the population. So what we mean by the population is if we sequenced everyone, we would find about 100 million different single nucleotide polymorphisms between everyone. Now, for one individual who's carrying one copy of their DNA from their mother and one from their father, there might be about 3 million SNPs that are actually present in that one person. So most SNPs only have two alleles, which means, again, if you sequence everyone in the population for a given SNP, typically what you would find is that everyone is either going to have a G or a T at this position. So it's not going to be the case that if you look at a bunch of people, somebody will have a G, some will have a T, some will have an A, and some will have a C. It tends to just be one or the other. So these are probably mutations that happen once in the course of human evolution and haven't really happened again. Now, just because you have two alleles does not mean that they occur at equal frequencies in the population. It doesn't mean that half the population is going to have a G at this position and half's going to be a T. Sometimes it could be 1-to-1, but also it may be more rare. So you may have certain alleles that are only maybe at 1% in the population. One of the things about single nucleotide polymorphisms is that they're very stable and very reliable. So this is in contrast to some of the other types of polymorphisms which tend to evolve more rapidly. They tend to change more. One of the types that tends to change a lot are microsatellites. So microsatellites are 1 to 3 base pair elements that are repeated over and over again in the genome, so maybe somewhere between 10 and 100 times. And these could just be long strings of one nucleotide, could be a dinucleotide sequence, a 2-nucleotide sequence that's repeated over and over, or a 3-nucleotide, and this goes on and on. So there are some with 4, some with 5. Most of them are 1 through 3. These occur fairly often in the genome as well, so about once every 30,000 base pairs, about 30 kilobases, there tends to be one of these microsatellite repeats. These, in contrast to the single nucleotide polymorphisms, which tend to only have 2 forms, can have multiple alleles with different numbers of repeats. So one individual might have, say, 34 of these A's in a row, another one might have 32, another person might have 45, another person might have 53, another person might have 18. So at a given location in the genome, you can have many, many different types of alleles. And we talked a bit about mini-satellites as well, and mini-satellites just have longer repeating units, but largely the same sorts of rules apply. So one of the goals then of modern genetics is to identify the genes that underlie disease. And today we're going to talk about 2 approaches, one of which is something that does not get used that much purely because we're not that lucky most of the time. So sometimes it's possible to identify a gene product, the protein, directly, and we'll talk about hemophilia as one of these examples. So instead of identifying the actual protein directly, most of the time we're going to be identifying the gene by linkage to molecular markers. And this is something called positional cloning, or map-based cloning, or chromosome walking. And this is nice because it's a much more general sort of technology. So as long as you can identify the phenotype, as long as you can observe the disease in a population, then you can identify the genes that underlie those diseases using this sort of a technique. So let's talk a little bit about the first approach. So hemophilia is an example of being able to clone a protein, identify a protein directly. And hemophilia is a typical X-linked recessive disease, as we've talked about before. You can see that in this pedigree, where the carriers are indicated with these little dots. It occurs mostly in males. So the reason that hemophilia is sort of lends itself well to this identification of proteins directly is purely because people have studied this pathway in some detail as to how blood clotting actually occurs. And people have a good understanding of the stepwise progression of different factors that are required in order to get blood to clot. And because we know what all these proteins are and we can detect them, it's fairly simple then to look in wild-type individuals where you can identify all of these factors, and then look in hemophiliacs to look and see which of these proteins are missing. So again, this works for hemophilia purely because the biochemical pathway, all the proteins have been identified, and we can detect those proteins and look directly for the one that's missing. So this gives you the protein sequence, or it gives you the protein that's affected in this disease, but it doesn't actually give you the gene sequence. So how do we get the gene? So we need to essentially work backwards to get to the gene sequence. So usually we think about screening libraries and finding the DNA sequences, and that tells us something about the protein that's involved, but you can go the other way around, where if you start out with the protein, you can work backwards to actually get the piece of DNA that encodes that protein. So factor 8, which is the one that's involved in this particular type of hemophilia, if you identify this protein, you can sequence the protein using amino acid sequencing, so you can actually determine the amino acid sequence. And then you can work backwards, since we know what codons encode which amino acids, you can work backwards from this protein sequence to get a DNA sequence. Now the only trick with this is, as we've talked about before, the DNA sequence, the codons are redundant. So there's more than one codon which would encode a given amino acid. So in this case, AAC, AAA, or AAT would encode this particular amino acid. So whenever we work backwards to get from the protein sequence to the DNA sequence, we end up with degenerate sequences, meaning that there are many different sequences which could have encoded this given protein. So this is fairly easy to synthesize, though, whenever we make these oligonucleotides. So when we go to synthesize a piece of DNA that will contain this sequence, what we can simply do is, when we're adding these sequences on chemically to grow this piece of DNA, we would add an A, then we would add another A, and then when the next base gets added, we would add T's, A, and C at the same time. So again, we're sequencing millions of molecules at the same time, so some of them would end up with a T as the next base, some would end up with an A, some would end up as a C. And then we go back to single nucleotide sequences with T's, T, G, G, C, C. So what you end up with then is a population of oligonucleotides which would all encode this particular protein sequence. And then these we can use as probes to screen genomic libraries or cDNA libraries like we've talked about before, and this will identify the actual pieces of DNA in the genome that encode this particular gene. So in this way, it's possible to work backwards from a protein sequence to actually get the DNA sequence. However, like I said, most biochemical pathways we don't understand that well. So blood clotting, failure for the blood to clot, is a fairly obvious phenotype, that if blood doesn't clot, then you have a problem in blood clotting. Something like cystic fibrosis, however, is much more complicated. So it's an abnormally viscous secretion of the lungs, pancreas, sweat glands, and other tissues, which has a lot of problems, especially for the lungs. For this, there's no real good idea of what type of a protein you're looking for, so positional cloning becomes an attractive option. And basically what we're going to do is just look for linkage between molecular markers and the phenotype. And the idea is the same one we've talked about over and over again with linkage, that the closer two pieces of DNA sequence are to each other, the more likely they will to be inherited together. And the further apart they are, the more likely they are to segregate independently. The advantages here are that this is a very versatile technique, so as long as you can observe the phenotype, you should be able to clone your gene this way. So with positional cloning, basically what we're doing, as I just mentioned, is looking for linkage between a marker and your phenotype. So the first thing to do is basically just to get some marker which will show linkage to your phenotype. So remember that if we're dealing with a human disease, you've got 23 chromosomes. So you need to get a marker that is at least on the same chromosome as your disease to have any chance of seeing linkage. And even if you're on the same chromosome, it has to be fairly close. So one of the first things you need to do is sort of a low-resolution linkage mapping, which is to try a bunch of markers and try to find a marker that's linked to your gene at all. And then once you find that will kind of tell you what section of which chromosome your gene is on, and then you can proceed with what we call high-resolution mapping, which is not much different than low-resolution mapping, but just trying a lot of additional markers in this region to find genes which show tighter and tighter and tighter linkage. And once you get down to a very small region, which may be down to something like 1,000 kilobases, maybe 1 centimorgan, this will still contain maybe 15 genes. So this is a big advance over the whole genome, which has 25,000 genes. So getting it down to 15 is a pretty good whittling down to a short list. But nonetheless, there's still some work to be done in identifying which of these 15 genes are really responsible for your phenotype. So low resolution mapping. So you just want to know where is your gene in the genome? What chromosome am I on at least? So there's lots of molecular markers to choose from in the genome. So we talked about, there's millions of SNPs and tens of thousands of single or satellite markers. So it's fairly easy to find lots of markers all over the chromosomes. And again, we're looking for linkage between the phenotype and the molecular marker. We'll talk a little bit about what this means in a second. So how many markers do you have to look at to actually find linkage? Oh, remember that linkage is based on recombination frequency. So if you look at the map of the whole human genome, it's about 3,000 centimorgans. So how many markers do we need to cover 3,000 centimorgans? Well, if the distance between loci is greater than 50 centimorgans, then you won't, if it's less than 50 centimorgans, then you'll see linkage. But if it's greater than 50 centimorgans, they'll be unlinked and they'll segregate randomly. So maybe this isn't not an etched in stone sort of rule, but markers about every 30 centimorgans is a pretty good place to start. And that would mean about 100 markers to cover the genome. So if you have markers every 30 centimorgans, what's the furthest that your gene can be from a marker? Well, if two markers, say M3 and M4, are 30 centimorgans apart, then the furthest your gene could be from a particular marker would be right in the middle, which would be about 15 centimorgans. And 15 centimorgans is pretty tight linkage, so linkage should be fairly obvious. So if it's anywhere not straight in the middle, if it were over this way or over that way, you'll be even closer to one of those markers. So this should give you a pretty good chance of seeing the linkage. Alright, so how does this actually work in practice then? So this is where marriages between family members become very useful. So human beings are outcrossing, so we don't self-fertilize very often, which means that most of our genome tends to be very heterozygous. So you got one set of chromosomes from one parent, one set of chromosomes from the other parent. Those are going to have lots of single nucleotide polymorphisms, lots of microsatellite differences, and so they're very dissimilar. They'll have lots of polymorphisms. But if you think about what happens whenever you have a marriage within family members, so if we have a marriage back here, gave rise to a couple of generations, and then we have a marriage between these family members, then a lot of these offspring are going to be segregating for the same sorts of alleles which were present back here. And in this case, if there was somebody who had cystic fibrosis back here, and now these parents were both carriers, and many of these offspring now have cystic fibrosis, we know that they're probably carrying the same allele. So they are probably homozygous for the same mutation that this parent carried way back here. So what does this mean then? So if we look at all these offspring, we have offspring which have cystic fibrosis, and we have many that don't have cystic fibrosis. So what we can say is that all of the ones that have the disease must be homozygous for that recessive allele, while the other ones are not going to be homozygous. They're going to be heterozygous, or they could be homozygous wild type. We don't really know, but we can say that all of the afflicted individuals are homozygous. What this means then is if you think about scanning through their genome, going over the chromosomes, that whenever you get to wherever this gene is in the genome, that every one of these individuals will be homozygous. Therefore, if you look through all these individuals and you just kind of go start scanning across the chromosomes and ask, are these segregating randomly for alleles on the chromosomes, or are you seeing linkage and they're all homozygous? Then this will tell us where these genes are. So just kind of to put some actual data to this, or some virtual data. So if we take a molecular marker, say it's on this chromosome marker 1, so number 1, and what I'm showing you is sort of an A and a B to show you the two different forms of whatever allele we're looking at. So marker 1 could be a microsatellite marker, where allele A has more repeating units and gives you a larger fragment, where allele B gives you a smaller band which has less repeating units. So this is just telling the two chromosomes apart. The maternal chromosome and the paternal chromosome are going to have a difference at this particular marker, and we can see that. So what we can see then is we know that, again, all these individuals in black have to be homozygous at the position on the chromosome where the cystic fibrosis gene is. And what we see is that we're seeing these A alleles and B alleles pretty much randomly. So they're not showing any real linkage, because if they were tightly linked to this, then they should all be homozygous as well. And we're seeing some homozygous A, some homozygous B, some heterozygotes. We're not really seeing much evidence of linkage. So if we kind of start walking across this chromosome, if we go to the second marker, same sort of an idea. We can just tell those two chromosomes apart. Then we can see also that there's a fair assortment of C's and D's. So we're not really seeing tight linkage with this disease-causing locus. If we go a little further over to M3, now we kind of look at this and say, okay, well, there's a lot of ones which are homozygous for E, and there's some that are homozygous for EF. So maybe we're seeing more E alleles than we're seeing F alleles, so maybe we're starting to see a bit of linkage, but it's not very tight at any rate. So if we keep going over to 4, now linkage is starting to look pretty tight. So given that there are 2 different alleles here, almost everyone who's homozygous for the cystic fibrosis gene is also homozygous for this G allele. So now we're starting to see some linkage. So we know that marker 4 then is getting fairly close to wherever the cystic fibrosis gene is on the chromosome. And if we go over one more, so we saw essentially no linkage with M1, 2, maybe we saw a little bit of linkage with 3, now we're definitely seeing linkage with 4, which indicates that the gene is somewhere over this way. So if we try with M5, marker 5, we see now that everybody who has homozygous for cystic fibrosis is also homozygous for this marker I. So this tells you then that this gene is very close to this marker. Now if we go even a little bit further to M6, we start to see a few recombinants again. So we start to see a few that are heterozygous, so they're not all homozygous anymore. And what this means is we've probably gone past the gene. So the gene is probably somewhere in the vicinity of M5. We saw a few recombinants on this side, a few recombinants on this side, so the gene is likely someplace right in the middle. And then basically, once you have done sort of your low resolution mapping and kind of gotten an idea where this gene is, then you can basically just get more markers. Because again, those SNPs and those microsatellite markers are very abundant, so we can find lots and lots of markers in between. And with any luck, you can get this interval down to about 1 centimorgan, or maybe even a bit less, which would be about 1,000 kilobases. And this kind of shows you some sort of a higher resolution version of what I was trying to show you, where if you find that a gene is between 2 particular markers, then you can go into that region, identify lots of new markers, and test those on your population as well, looking for where those recombinants are, and identify a set of candidate genes that are in between. So what about identifying the genes then? So, after all, you've done all this linkage analysis, but there's still on the order of 1 million base pairs of sequence in between your 2 closest markers, containing somewhere between 10 and 20 genes. So how do you figure out which gene it actually is? Well, one of the most straightforward things you can do is to sequence wild-type and diseased individuals. So, if you find something really obvious, that all of the wild-type individuals have a nice open reading frame that should encode a normal protein, but you find that the diseased individuals have, say, deletions in that gene, they have insertions in the gene, they have frame shifts, some really severe sort of mutation, then that is a pretty good piece of evidence that you've found the right gene. So this is possible. It's actually not so expensive anymore, but it might still not give you a definitive answer. So in some cases, anyway, when you sequence the gene of interest, you might find there's only a missense mutation, that there's just one amino acid change. And then it's tough to say whether that one amino acid change is just simply a naturally occurring polymorphism in the population, or whether that could really be a difference that is actually causing the mutant phenotype. So there's a number of sort of pieces of detective work you can do to try to get some sort of an idea of which of those 10 to 20 genes in your interval might be the right gene. And looking at the RNA expression patterns of those genes in the interval is a good way to sort of get some information. So the expression pattern of a gene often gives you some idea of what its function is. So one of the things you can do is northern blots with various tissues. So again, all cells in your body have roughly the same DNA, but they're not using all of the same genes. So some genes are going to be only expressed in particular tissue types, whereas other genes may be expressed in many tissue types. So if you collect RNA, what's being expressed in different tissues, say ovary, testes, lung, and blood, purify the RNA, run it out on a gel, transfer it to a membrane, and then probe with radioactive pieces of DNA corresponding to your gene of interest, then this gives you an idea of where those particular RNAs are being expressed at. So in this case, you're looking at a transcript that is specific to the testes. So what does this tell you? Well, if you're looking at a disease like cystic fibrosis, which affects many organs, then this sort of an expression pattern of a gene which is only expressed in testes, and say not expressed in the lungs, where some of the main mutant phenotypes in cystic fibrosis are, this is probably not a good candidate. But say if you were interested in a phenotype which occurred in the testes, then this would be a good candidate gene because it's expressed very highly in the testes and not in other tissues. So just to kind of go with this northern blot idea for a second. So you can get a lot of information out of the northern blots. So what I just showed you before was trying this with different tissues, and this would tell you where this particular gene was expressed at in terms of what tissue. You could also do northern blots with wild-type individuals and individuals which show the disease that you're interested in, say mutants 1, 2, 3, and 4. And if you probe with a given gene, what you would find is you can find lots of different pieces of information. So in wild-type we see that we get a certain band of a certain size and a certain density. So how dark these bands tell you something about the expression level of the gene, whereas the position up and down tell you about the size. So smaller fragments are at the bottom, larger fragments are at the top. So if you compare wild-type individuals to mutant individuals, you could see various things. Like here we have one where we have a lower than normal expression of this gene, or we could see a higher than normal expression of this gene. So either one can lead to a disease state, underexpression or overexpression. We could also see differences in size in addition to the abundance. So things which have a larger than normal size, these could be mutations that contain something like an insertion. They could also have a mutation in the splice junctions, so that maybe an intron can't be removed now and you're making a larger than normal product. Or you could have individuals that say contain a deletion in this gene, or a premature stop codon which would make a smaller than normal product. Similarly, you can do the same sorts of analyses on proteins if you are lucky enough to have an antibody to this given protein. And this is called a Western blot whenever you do the same sort of analyses with proteins, but essentially it gives you the exact same piece of information. You'll get the size of the protein, the abundance of the protein, and this can tell you something about whether these mutant individuals have had some sort of a problem in the expression or the processing of this particular protein. So finally, confirming candidate genes. So once you have a candidate gene, then what? So we can do sequencing to see if there's a mutation in it. We can sequence wild-type and diseased individuals. If you're lucky, you'll find that all the diseased individuals have mutations and all the wild-type ones don't. Sometimes, though, it can be a little confusing. So if you sequence 2 individuals with cystic fibrosis, you're very likely to find that they don't have the same mutations. So all of these little symbols below the gene here show different mutations that have been identified in people with cystic fibrosis. So what we can see is there are many, many different mutations that lead to cystic fibrosis. So sequencing may be a little bit inconclusive in that if you sequence 2 individuals with your disease and one has a stop codon and one has a missense mutation, or one has just a base change in the promoter, it may be a little tough to completely conclude that that's what's causing your phenotype. So what you can do, so sort of the gold standard for definitively showing that you've identified the right gene that's causing your phenotype, you can do this in 2 ways. So one is to actually recreate the phenotype. So to create a new mutation in your candidate gene and see if it has the same phenotype. Now obviously this is something we can't do in humans, so you can't deliberately take a wild-type human being and knock out a gene to see if they get a disease. We can, however, do this in mice. So we'll talk later in the course, towards the end, about how you do this technically, but you can take the gene in mouse, so we could take that cystic fibrosis gene in mice, and deliberately knock it out. And if when you knock it out, you get that phenotype, you get the cystic fibrosis phenotype, then this is proof that you've identified the correct gene. The other way you can do this is to go just the opposite way around. So you can rescue the mutant phenotype, and this is usually done in mice as well. And in many cases, we have models for disease in for many diseases in mice. So there are mice which get various types of cancers and diabetes and are overweight and have all sorts of different diseases. And if you think you've identified the gene that's responsible for that disease, then what you can do is transgenically put back in a good copy of that gene and see if it restores a wild-type phenotype. So this rescuing the phenotype, putting back in a good copy and looking for a restoration of the wild-type phenotype, is another definitive way to show that you've identified the right gene. And again, we'll talk a bit about the technicalities of how you do this later in the course. So just real quickly to kind of walk through one example of this using northern blots to identify which genes might be responsible for your disease. I just want to walk through one example of this. So let's say you're positionally cloning a gene that's involved in heart disease. Disease does not show any symptoms in other organs. So what this means then is this gene is either it's only expressed in heart or at least it's only critical in the heart. So let's say you've done your positional cloning and you've got down to a region that contains 5 what you think are genes. So you perform northern blots on each of the 5 genes. On tissue that's been isolated, or RNA that's been isolated from liver, heart, and muscle. So basically what we have here are probes, so we've made probes to these 5 genes, A, B, C, D, and E, and we've used them to probe northern blots made from RNA, from liver, from heart, and from muscle. So just kind of to ask some questions just to make sure that everybody understands how this works. So which regions contain a gene? So we have these genomic regions A, B, C, D, and E. So which contain a gene? Well, the only evidence we can really get from this particular experiment that there is a gene there is the fact that you can detect a messenger RNA. So with probe A, we detect a messenger RNA here and here. So region A contains a gene. We detect transcripts with C here, and we detect transcripts with E here. So what we can say is that regions A, C, and E definitely contain genes. So what can we say about regions B and D? So they don't detect anything. So does this mean that they don't have genes? Well, there's a saying that the absence of evidence is not evidence of absence, and this is a good example of it. So we can't really conclude that there aren't genes here. We just haven't seen any evidence yet that there are genes. So if there are genes, why can't we detect them? Well, we've only looked at 3 different tissue types. So maybe regions B and or D contain genes that are expressed in, say, neurons, or in skin cells, or in the immune system. So if that were the case, then these things might very well contain genes that are expressed in other tissues. All we can really say right now is that B and D don't obviously contain genes that would be expressed in liver, heart, or muscle. So how many genes have been identified? I would say 3. So A is detecting a transcript here and a transcript here. They're about the same intensity, they're about the same size, so we'll assume that those are the same gene. Same with C and E. So we could say that in this region, there are at least 3 genes. And which fragment is most likely to be responsible for our disease? Well, our disease said that it was something which was expressed in heart, a phenotype that was exhibited in heart tissue, but not other tissues. So we could see that gene A is not expressed in the heart, so this is probably not a likely scenario. Gene C is expressed in heart, but it's also expressed in liver and muscle. So you might expect that if you knocked it out that you would see phenotypes in liver and muscle as well as heart, which is not what our disease is. But gene E, since it's only expressed in heart, then this is the most consistent with that model, that it's something that is important to the heart but not other tissues. So, it's not completely definitive, but at least the best guess, giving this particular set of data, is that probably gene E is the one that's most likely responsible for that particular disease. Okay, and that's all for today.